All right, let's get right into it. We're going to be digging into a fascinating scientific investigation, one that uncovers a silent contamination, a hidden danger that's lurking on the very food we think is the healthiest.
You know, when you see fresh, vibrant produce, you're thinking health, right? Nutrition. But what if I told you there's an invisible threat hiding right on the surface of that tomato or kiwi, something you can't see, you can't smell, you can't taste, but that poses a massive, growing risk to our global health? So, what is this threat? Well, it all comes down to these antibiotic resistance genes, or ARGs for short. Think of them like tiny genetic escape plans for bacteria. When a bacterium has one of these genes, the antibiotics we count on to fight infections, well, they just become useless against it. This is exactly how superbugs are made. And the scary part? Scientists are finding these genes in some really unexpected places.
Okay, so how do you even begin to find an enemy you can't see? Well, scientists basically had to become detectives. They started this massive genetic investigation, kind of like a high-stakes detective story, where they needed to crack the code of the bacteria living right on our plants.
And when I say massive, I mean massive. They analyzed the complete genetic blueprints. We're talking the entire genome sequence of over 1,000 different bacteria, all of them sourced from plants. A scale like that gives their findings some serious weight.
So let's meet the key suspects they found. You've got genera like Streptomyces, which is really common in soil and on potatoes. Turns out it had the highest average number of these resistance genes. Then you have Pseudomonas and Xanthomonas. You find those a lot on crops like tomatoes, and they were also carrying a pretty significant number of these ARGs. What does this all mean? It means the bacteria living on some of our most common foods are literally carrying the genetic codes for antibiotic resistance.
Okay, so we know who is carrying these resistance genes, but the next big question is, how are they spreading? This is where things get really interesting because we're about to look at the superhighway for antibiotic resistance. Meet the main villain of our story, the conjugative plasmid. The absolute best way to think about this thing is as a getaway vehicle for genes. It's this small circular piece of DNA that can just copy itself and then zoom from one bacterium to another, carrying all its genetic cargo, including those really dangerous antibiotic resistance genes, along for the ride. And this diagram, it shows you exactly how it works. It's basically the blueprint of one of those plasmids they found. See that little red bit? That's the weapon, the antibiotic resistance gene itself. And all those blue parts? That's the engine. It's all the genetic machinery that lets the entire plasmid move between bacteria. So the really crucial thing to get here is that the weapon comes pre-packaged with its own delivery system. So now that the scientists had identified the getaway vehicle, they could start tracing it back to its source. They started following the genetic trail, trying to figure out where the most dangerous, the most mobile of these resistance genes were actually coming from. And this is where they made a really critical distinction. On one side, you've got your soil-native bacteria, like Streptomyces. Yeah, they have resistance genes, but they're mostly built in. They're stationary. They don't really travel. But then, on the other side, you've got these crop-associated bacteria, like Pseudomonas and Xanthomonas. Their resistance genes, they're often acquired, and they're riding on those high-speed getaway vehicles, which makes them a much higher risk for spreading this resistance around.
So, this brings us to the million-dollar question, the one this whole investigation was leading up to. If these high-risk genes are being acquired by the bacteria on our crops, where on earth are they getting them from?
And the study points to one very likely source, livestock manure. You see, the widespread use of antibiotics in animal farming creates this perfect breeding ground for resistant bacteria. So when that manure is used as fertilizer, it's not just introducing nutrients into the soil, it's introducing a flood of these highly mobile, really high-risk resistance genes into the agricultural environment. And from there, they can be picked up by the bacteria living right on our plants. Now look, uncovering a problem this big can feel a little overwhelming, right? But the great thing about science is it doesn't just identify the threat, it also points us toward a clear solution. So this final part of the story is all about how we can actually break this chain of contamination.
And the authors of the study, they lay it out for us directly. They propose a really critical point of intervention. Basically, instead of trying to clean up the mess after the fact, they're saying we need to act before it even begins, by treating the manure before it ever gets spread on the fields. And here's their plan. It's a pretty straightforward 3-step approach. Step 1, monitor the manure for these mobile resistance genes. Step 2, if you find them, remediate the manure. Clean it up, neutralize the threat. And then, and only then, step 3,
use that treated manure to fertilize crops safely. It's a way to effectively cut off that resistance highway before it ever reaches our food.
So what this investigation really shows us is that the microbiome of our plants, the tiny world of bacteria living on them, is a critical link in this huge global network of antibiotic resistance. It connects the farm to our fork in ways we probably never even imagined. And it leaves us all with a really vital question to think about. Knowing that the safety of our food truly begins in the soil, are we doing enough to protect it?